Now it's time for the Director of the Office of Cancer Clinical Proteomics Research at the National Cancer Institute in the US. It's Henry Rodriguez. He has been instrumental for our work establishing Moonshot in Sweden too. We have visited him several times, and he has been doing a lot of things to ease our work in promoting Moonshot. So please, Henry.
Okay, thanks, Georgi. Can people hear me there?
Yep.
Okay, perfect. So first of all, let me just say that, yes, Georgi is correct. It's the second invitation that I've had to turn down, but this one's quite special that only a couple of people know, and Georgi is one of them. My daughter just graduated from college. This week is graduation week, and my family is celebrating here in Washington, D.C. prior to her going to law school. So I apologize for not being there, but I promise next time if there's an event and if I have availability, I will definitely make it a trip to go and see everyone. So with that, what I've been asked to do is to give sort of an overview of what we've been now doing at the Cancer Institute, and more specifically, how we're starting to see cancer as something that you begin to understand at a biological level. But we see that from where technology has been, where it is evolving, and how we begin to mix those worlds together. The other aspect that I will briefly touch upon is a new effort that we've launched, which really begins to capitalize on the mere fact that cancer is a global issue. And we're trying to work collaboratively here within the US and as many countries as we can to ultimately put an end, or at least an understanding, to this deadly disease. And within that, I'll touch briefly about an effort that we've now launched here just about 2 years ago in coordination with the prior administration in our White House. So a little bit of science background, but I'll keep it at a very high level. I think everyone in that room, if I tell you you're going to go to a hospital and you're going to take a genetic test, you kind of understand what that means. You might not understand it scientifically, but you believe it's something that we here in hospitals could administer to a patient to ultimately better understand the disease, try to understand how you're going to respond to treatment, and potentially determine exactly which treatment to administer to you. And one of the things that attracted me 12 years ago when I came to Washington, D.C., and I got hired at the National Cancer Institute, is that it's an organization that we enjoy taking risks. And I'm one of those that firmly believe by taking risks, ultimately you could see science and patient care move forward. One of those large initiatives that started this revolution of genomics, as we refer to it, actually got created approximately in 2006. And it's a program now referred to as the Cancer Genome Atlas, or TCGA. And this is a program that has done in the past 10 years an incredible feat. It has gone through approximately 14,000 individuals comprehensively, meaning you can measure as many things as you possibly can using your technologies, and we want to understand it at the genomic level. And we looked at about 34 cancer types. But at the same time, we also understood from an institute that genomics is not all that you should be looking at. There's more in biology. And let me explain it to you. Genomics people typically refer to as the blueprint. Imagine if you're building a house. You all have a blueprint if you want to build your home. But ultimately, there's going to be workers that will develop your home. Sometimes the blueprint does not turn out to the final product. It is critical to be able to not only measure, but to understand what those workers are, what they're doing, and how it affects the ultimate product at the end. That is the science of the measurement of proteins. Because of that, the National Cancer Institute, when they created the Cancer Genome Atlas, they also decided to comprehensively characterize not just genomics, the genes, but the proteins themselves, the science that we refer to as proteomics. The program that they created for that now affectionately is referred to as CPTAC. But they did something very unique here at the NCI. Because of some manuscripts and some science claims that were made earlier, they kind of shed sort of a dark cloud on the science of proteomics. The Cancer Institute decided, we believe from a technology perspective, genomics is very mature. So they went after biology for the Cancer Genome Atlas. But when it came to the measurement of proteins, we took a very conservative approach. And the reason for that is twofold. One, it is critical not only to measure the proteins themselves for biology, But you need to be very sure of what you're measuring, because ultimately, almost every drug that we administer today to a patient targets a protein. And that's key for us to understand exactly what dose to administer to a subject and to determine whether or not the dose is actually hitting the exact target that it should to better patient care. So as I said, what we wanted to do was to understand the technology. So we first tried to understand and standardize as much as we can. That was the first program. Once we understood and we believed wholeheartedly that through standardization, proteomics was a mature technological science, then we decided to go after the biology. And for the biology, we did not want to treat the measurement of proteins in isolation. We wanted to pair it with our sister program, the Cancer Genome Atlas. So what we did, we specifically took a patient sample that already underwent characterization through the Cancer Genome Atlas genomically, and then we wanted to measure the proteins within that patient's tumor. And ultimately, we're simply trying to ask is, are you able to gain a better understanding of the underlying biology of the patient, and subsequently potentially use that as a hope of moving the field forward? So we went after 3 cancer types. I think we were very fortunate within this pilot program of the Cancer Institute. We studied colorectal cancer, breast cancer, and ovarian cancer. The overall highlights made it very clear to us. In each one of these instances, when you pair the measurement of the proteins, the science of proteomics, above genomics, you identify new biology. Furthermore, you identify potentially in cancer types, new subtypes. And this is key as a way we try to identify and try to stratify our patients on which treatment to administer to them. And furthermore, they had better correlations with the clinical outcomes of those cohorts themselves. The other thing that was quite, from a biological perspective, people typically, historically, will always use genomics to infer computationally what the proteins are, what their quantity would be, and what they're actually doing. And we showed that while it does have some merit, overall, it's not the best thing to do. To physically measure those materials plays a key role. Overall, what we learned, pure and simple, your ability to measure not just your genes, but to couple that with the proteins themselves at the same time, we're able to gain a better overall picture of the biology of cancer within our patients. So this was good news. Furthermore, it allowed me to go back to my board here at the Cancer Institute and issue another iteration of our program over the next 5 to 10 years. But this actually caught the eye and the attention of someone very important that was in the White House a couple of years ago. Specifically, that was the 44th or the 47th Vice President of the United States, Joe Biden. If people are not familiar, in 2016, under leadership and the inspirational words that actually, the call that came out by Joe Biden, he created what's now referred to as a cancer moonshot. But what caught me and really drew me to this was the simplicity of the overarching goals. And there's 3 of them. One, how can you develop ways, mechanisms to accelerate the progress in cancer? There's many ways that you could kind of answer that question. But the last 2 are very dear to my heart, and it's the way that we've run the US-based operation. And that is, can you develop vehicles, tools, mechanisms to encourage greater cooperation and collaboration, not just within a university, because I think everybody does that, but across institutions, and more specifically, across countries, if that would be possible. And if you were to achieve that, can you work with them and hold their hands and convince them in a way to not to hold the data that they generate from their research, but ultimately to give it back to the public, share it with the globe as a whole. Because we've learned if you keep the data within yourself, sure, you're able to extract valuable information, but ultimately if you allow other people to look at those data sets, new information, new eyes look upon it, and potentially new discoveries can be identified. And quite frankly, I have never yet met a patient that says, I want to keep all my information private. I want to give it up to the world, so hopefully greater discoveries come out of it. So because of that, I started asking a bunch of what ifs approximately 2 years ago. And we started asking this vision, could something be created? And that was ultimately, can you create a forum? A forum where people would be free to collaborate. You bring in the leading institutions that either would treat historically genomics individually or proteomics individually, And now, because of the science that we're seeing, bring those two worlds together. The cancer types, we would work with each of these nations. They would be selected, and the funding for those projects would be by each country, bringing significance to the leaders within each one of those nations. Because here at the Cancer Institute, we spent a lot of time trying to standardize the various workflows to ensure that what you measure is going to be representative of biology, not an artifact of the way that you're doing the experiment. We wanted to adopt as many of these as you can when appropriate. And again, the key for us is that they would sign what we refer to now affectionately as a data sharing pledge. So this was the pitch that we gave to the White House. Ultimately, it caught the attention of the White House itself, and I get a call back. And the question then becomes, very good, can you start building this, and are other countries going to be interested in developing such an effort? So we tested it out. The very first country that signed on board was Australia. That country came on board in July of 2016. It was a great momentous occasion. The former vice president flies out to Australia. He does this kickoff event, and he even utters the words that we would love to hear here as cancer researchers. He uses the words proteogenomics, which is the merging of those two disciplines. He even talks about proteins. But he also said one thing that at the time, I did not quite comprehend what that would imply. And he said, I predict that you're going to see this type of collaborative effort replicated or repeated around the world. Now, to be very clear, Australia got launched by the individuals that are listed at the bottom of the slide. They played a key role in believing and taking this risk. And a huge thank has to go to my colleague, Mark Baker, which really worked hand in hand with me to pull this off. But what did exactly did the vice president mean by that? Well, what that meant was when they came back to the United States, I then get another phone call. And the question becomes, well, we loved it. Can you replicate this now in other countries? More specifically, if you can, you have 8 weeks to do this. Because something might occur in New York City that we would like to create an announcement. I pretty much had a heart attack, to put it in a very good way, but I took it as a great challenge. Because I myself, I happen to have had a mother, if people are curious, who has suffered from breast cancer. Luckily, she's a long-term survivor. The past 2 years, I've lost 3 relatives, and that is a reason why I tend to fly a lot, because I'm spending my time now going to various hospitals doing these referrals. But that said, I felt this would be a wonderful opportunity to kind of build this sort of an effort, if you can, across many countries. So we took the challenge. To our surprise, it resonated. At this point, in the span of 8 weeks, then we went from 1 country, 4 institutions. We now signed on 7 additional countries, and we signed on 10 additional institutions. We all got together in 2016 in Taiwan. There was a large conference that was occurring, and we all started this now tradition that we hold. We hold our representative country flags out of the sense of pride. It was a huge pride for me because I also had the opportunity, as the conference was held in Taiwan, to meet and have a very cordial dialogue with the vice president of Taiwan. And of course, the reason September, I later on found out, was very critical because the very next day, Joe Biden announced all these partnerships at the United Nations Foundation Social Good Summit. So this was September of 2016. Obviously, we're now in May of 2018. The question now would become, so what's come out of this? This has taken a life on its own. Today, this is now an official structure. It is now affectionately referred to as ICPC, the International Cancer Proteogenome Consortium. It now consists of 12 countries representing 31 institutions, all united in the belief that converging the disciplines of genomics with proteomics is going to provide a tremendous acceleration in our understanding of cancer biology and potentially move that towards patient care. And again, to be clear, all these institutions, all these countries, they have signed on what people said you could never do. They have signed a data sharing pledge where the data, the research data that they develop from their projects in coordination under this Moonshot effort will be placed in the public domain. They could host them in their own countries, but a copy of those data sets will come back here to the US, to the National Cancer Institute, where we will host it in perpetuity for years to come. Now, This is not the only program that we run that is beginning to merge these disciplines together. So as I said, CPTAC was one of the first ones that wanted to take this risk. International Cancer Proteogenome Consortium is another one that has now taken this effort. There's another effort within the United States involving the federal government, referred to as the Apollo Research Network. I believe Tom Conrad is in the audience. He could speak more to that. But this one specifically involves the National Cancer Institute, the Department of Defense, and the Veterans Administration. And there's other efforts and recommendations that has come directly from the White House to our institute here in the US, the NCI, that's been issued by the Blue Ribbon Panel, which was a task force that was brought forth by the White House. And again, all the information, all the data is going to be placed in the public domain. Because I have learned that while my own program, we're able to analyze the data and put out very nice publications, other people looking at those data sets is able to solve additional questions that we might not have asked. So that's the opportunity that we want to capitalize upon. Now, lastly, I saw the patient advocate prior to my presentation give a very eloquent talk. I truly believe that one of the key things in moving science forward is not a scientist talking science. Quite frankly, I tend to fall asleep in a lot of these conferences too. I like it when the average person is able to convey the message to another individual. And the benchmark for me happens to be my parents. So using that as an inspiration, here at the Cancer Institute, we develop an educational video dedicated to the average person, to patient advocates, specifically trying to explain the science of proteogenomics. So let me give you a quick 2.5-minute clip so you could actually see that.
Proteogenomics research on the frontier of precision medicine. The fields that we currently consider next-generation molecular medicine, including genomics, proteomics, miRNAomics, microbiomics, and epigenomics, all share the same foundation. That foundation rests in omics, which is an informal term used to describe the comprehensive study of the biological components of a cell at the molecular level. Take genomics, for example. Genomics is a comprehensive study of the complete set of genes, DNA, in an organism, which we refer to as the genome. Analyzing the genome of a patient's cancer can reveal information about how to best detect, diagnose, and treat the patient. For example, a patient's cancer may have a DNA mutation that makes it especially sensitive to a drug. Although studying a patient's DNA can provide a lot of information, it doesn't give us the whole picture. The reason is that in addition to DNA, there are many other molecules that contribute to cancer biology, such as proteins. Proteomics is the comprehensive study of the complete set of proteins in an organism, which we refer to as the proteome. Proteins are built from DNA. You might say that DNA is the blueprint for life, and proteins are the tools that make living systems work. They play a major role in the daily functions of both healthy cells and cancer cells. Analyzing a patient's cancer proteome can also provide information about how to best diagnose and treat the patient. This is because proteins carry out almost all of the functions within our cells. If a protein is altered, a cell could begin to multiply uncontrollably, potentially leading to cancer. This is why almost every clinical cancer treatment targets a protein. Understanding how proteins influence cancer biology is critical to developing better diagnosis and treatment strategies, as they can add clarity to how a patient will respond to a particular treatment. In the past, researchers often studied patients' cancer genomes and proteomes separately. But in 2016, research teams combined the comprehensive analysis of patients' cancer genomes and proteomes. This integrated approach of proteomics and genomics is called proteogenomics. These studies showed that separately, genomics and proteomics provide a partial picture of cancer biology, but studying them together produced a more complete, unified picture. Researchers are hopeful that proteogenomics may improve our ability to prevent, diagnose, and treat cancer at the molecular level. Proteogenomics research on the frontier of precision medicine.
So Let me just leave people with some staggering facts. It's all about the patient, in my view. If you look at the statistics, simply within the United States, every year alone, just over 1.5 million individuals are going to hear the words you never want to hear. And that is, you have been diagnosed with some stage or grade of cancer. Out of that 1.5 million, just over 500,000 Americans die of cancer. To be very clear, this is not an issue that faces my country. This is a global issue. Cancer has no boundaries. On an international level, the latest numbers, on average, over 14 million individuals are going to be diagnosed with cancer. And on a global perspective, annually, over 8 million individuals die per year. To me, where I see the science of converging genomics with proteomics, what we refer to as proteomics, what Georgi and all the colleagues of mine there are doing in the audience, is ultimately we're trying to seek the latest technologies and we're trying to offer hope towards patient care. So with that, I want to say thank you very much, everyone.
Thank you very much, Dr. Rodriguez. I would just like to mention the corresponding numbers for Sweden. We have approximately 60,000 new diagnoses, cancer diagnoses every year, and approximately 23,000 people dying from cancer each year. And due to the increase in life expectancy, in 20-25 years, we will probably have around 100,000 new cancer diagnoses made every year. So this is a very important issue for all of us. Now the next speaker is Chistoph Borchers from Victoria, Canada. He's a professor and director at the Department of Biochemistry and Microbiology. Please, Chistoph.
So first I need to apologize that I'm late, but I was in the clinic garden, but in a different city. The city is called Hellingsborg, so 40 miles away. And the funny thing is that I called Georgie, I cannot find it. I stand here beside the local person, like maybe you can tell him in Swedish where I need to go. And they were talking, and I was like, okay, we need to round the building. Like, and we were walking around the building, and still the wrong city. And it took us like another hour to figure out we are in the wrong city. Well, anyway, I made it. So I want to talk a little bit about what we are doing in, how do I switch the slides here? In Canada and Germany. I'm one of the consortia of the Proteogenomics initiative initiated by Joe Biden, as you have heard from Henry Rodriguez. I don't know exactly what else you have heard today. Again, like it took me a while. I told you it took me a while to get to this place. But I wanted to mention this slide here over there. Where you can see like a lot of people are doing genomics. Here the DNA, but going from the DNA to the phenotype, there are other molecules in there which are also quite important. Do you know what a caterpillar is and what a butterfly is? Do you know what the difference in the genome is? Zero. It's the same genome, different phenotype, right? And because of these molecules in between. And we are talking about here the proteins. And you need to consider not only the abundance of the proteins, all the other things were shown over here, isoforms, localization, interaction, and so on and so on. There's many, many more things. I don't want to mislead you. Genomics is great, has done a lot of good things, but it's limited. Because from here to there are DNA, the RNAs and there are proteins and metabolites and even others. So coming back to our consortium. I'm here, I actually have two sites where I am. I'm at the University of Victoria in British Columbia on the island, which is called Vancouver Island. And in our consortium are the folks from the University of British Columbia, so Vancouver, our neighbors. And then my second side, I'm also working at McGill, is involved. And this over here, it's called ISAS. I know if you spell it out in English, ISIS, it's not so, can have a different meaning, but it's an actual institute, it's 60 years old, and it's in Dortmund, where my favorite soccer club is, BVB Dortmund. And that's one of the reasons why I'm working with them, but they're also very good in analytical science. So we have these 4 hubs. 2 of them, they're more focused on technology development. This is in Victoria and in Dortmund. The other one are really, this is the clinical folks, provide us with the samples. That's at UBC and McGill. So, and one of the things what we want to do is we want to develop assays for clinical research and as well as basic research. And this better? Okay. And as you know, like the most favorite animal model is mouse. And I'm so lucky that I got funding from Genome Canada to develop assays for around 3,000 proteins in mouse at different tissues and biofluids. And I do this together with the folks from Dortmund. And as you know, right now, the new technology, CRISPR-Cas9, where you can knock out and knock in a gene, but then you need to phenotype this again. And you look at the mouse, look at the fur, is the mouse blind or cannot hear correctly, but that's not everything. What is going on with the proteome? And here with these assays, what we're going to develop, we can check out the proteome in different tissues. And this is our status right now. We want to do 3,000. We have so far 1,200 assays developed, and focus on mainly on plasma, because plasma is, as you know, the main clinical biofluid. But even of other tissues, we want to consider lung. As you know, lung cancer is a big topic, and heart, and so on, liver. So this is one thing. And also, like, we want to focus 3,000 on not all the proteins. In mouse, we have at least 16,000, but we need to start somewhere. And this is a project going over 3 years. But what we want to focus on these proteins which are involved in the signaling pathway of cancer. Here, this is like a snapshot of the signaling pathway, and everything what is in yellow, we have already developed assays for. Another thing is like not only we want to provide technologies and methods, approaches for analyzing the mouse model, but also we want to analyze directly clinical samples from patients. One of the approaches we're doing is we having a comprehensive proteomics analysis and genomics analysis. And then when we have in both, we want to put this both together. It means like we are directing the protein analysis through the genomics results. One of them is over here. This is like a tissue from a patient of colorectal cancer, and that has been detected by the genomics analysis and a mutation in KRAS. And now we wanted to, if it is in genome, it's fine. It tells me it can be expressed, but doesn't need to be expressed. And now we want to see, like, does it really have, but did they make really the protein out of it, and how much is really there? Because if the protein is not expressed, who cares? And so we were able to analyze in a comprehensive way the proteome of this tumor tissue, and found the mutation in KRAS as predicted by the genomics analysis. And then we have developed assays for quantitation of these mutation, and interestingly, we saw like differences. The genomics analysis, it's around 30%, and we said at least 50% even more. Another way how we do this, like in a comprehensive way, in a semi-quantitative way, analyze or detect and quantify these proteins which are here in this pathway, and this gives us really a specific profile of each patient. Now we want to combine it with genomics and proteomics and artificial intelligence. I think this is really the next thing, like to be, because we are talking about 8,000 proteins over here, the genomics analysis, multidimensional. At least like I cannot do it. And like normally, like I said, is when you play chess, a good chess player can predict maybe 3, 4, 5 moves in advance. But here we're talking about multi-dimensional assays, which needs to be considered to be connected. So that's one thing. Another thing is like what we're going to do is like we want to bring in assays really into the clinical chemistry lab that's being used like not from research labs which are specialized in proteomics, pretty much from everyone. When you go into the clinic, they will analyze your proteome of this tissue in a very specific way. How they do this right now? They do this, but by this one, immunohistochemistry. It provides you certainly spatial information about the protein, but it has a lot of downsides shown over here. And it is subjective. You know the old joke, like you give a sample to 3 different pathologists and you get out 5 different answers. It's really very subjective. And even like I'm working at the McGill, it's very interesting, like I'm working with the chief pathologist together and say like, you know, this immunohistochemistry is very limited. We need to have something better. Why not doing mass spectrometry? Like most of the people are here, like they are mass spectrometrists, and this is like right now really the most promising approach, multi-reaction monitoring. It's fantastic, but it also has some downsides, and what I believe, it doesn't go into the clinic, in the clinical chemistry lab, at least right now, because it is too complex to learn, too not robust enough, and it's quite costly. And also, it takes too long. But what has entered the clinic already is MODMS, not right now, not for clinical diagnostic, but for microbiology. It has revolutionized microbiology. This kind of instrument, like this big, very simple, very robust, doesn't need anything, only a plug, no gases, nothing like, and runs. Like, what we think right now is when we combine immunoaffinity enrichment of the peptides of our interest and MOLDI, TOF-MS, as it has been shown in the microbiology, this could really revolutionize the clinic, and in particular in clinical diagnostic of cancer-related proteins. So how does it work? It's actually very simple. So you have the cell lysate from your tissue, like first you need to lysate this. This is when we are still on the protein level. You're adding some enzymes in there, shoes it up into peptides. Then we're adding, this is always what we do when we talk about mass spectrometry, we're adding an internal standard. It's the same peptide of our interest, but it's isotopic labeled. So it's a little bit heavier in mass. And this is what mass spectrometry is doing. It is not only determine accurately the molecular weight of, this case, of peptides, but also separating them based on mass. So then we're adding an antibody immobilized on beads. We wash it away, everything what is not specific. And then this is a trick, like then we're placing the beads directly on this plate and put it into the mass spectrometer. First, we're adding a matrix, which then eludes the peptide from the antibodies that can be measured. And then we shoot it with a laser. And what we're getting are 2 peptides in our MS spectrum. One at the lower M/Z corresponds to endogenous peptide, and at the higher M/Z, higher mass, corresponds to the internal standard. And based on the ratio, we can quantify. It's really that simple. So what we combine here is the peptide enrichment by antibodies, which gives us a high sensitivity, and the specificity comes from the mass spectrometer. The instrument, as I mentioned before, is very simple. And then the sample preparation, very important. If you bring something into the clinical chemistry lab, everything needs to be totally automated. And this can be done by liquid handling systems, and I will show you this in a few seconds. And it's very fast. With MIM, normally analysis can take, we do this like in 40 minutes. We're quantifying 250 proteins in 40 minutes. Well, it's not bad, but this is not really doable in the clinical chemistry lab. This goes like within 5 to 15 seconds, you have the results. And we think this technology can be used for biomarker validation, routine clinical analysis, and companion diagnostic. And what I mean with companion diagnostic is that you have some markers, and based on the quantity of these markers, you decide what therapy is the best. And this is where we really want to go, which is called personalized medicine. We have developed now an assay using this technology for this protein. There are other proteins in the signaling pathway. They have a lot of mutations, and patients getting stratified based on the mutations, but not for this major player, AKT, because it has just one mutation, and it happens only to 5% in patients. Then we're working here like with AstraZeneca together, and like they came to us and like, what we really need to know, we have an AKT inhibitor. This is an important protein. If we shut this down, we can cure cancer. So, but we don't know how can we stratify the patients. Like, can you provide us an assay which can do both? Determine the expression of the AKT and the phosphorylation. A specific phosphorylation which is involved in the function of the protein, which is shown over here. This one. And we do this right now for AKT1, AKT2 is actually another isoform, AKT3. But we have developed using the immunomoly technology and using right now immediately like an instrument which has been used in the clinic in the microbiology labs. And this is like what we do, what we have done so far. We can quantify AKT1 and 2 out of 100 micrograms total protein. Actually, right now we can do it at 10 microgram, and you can see like we can quantify the proteins out of cells, cancer cells, and also out of tumors. And now next step is really like to automate the entire sample preparation using a liquid handling system. One of them is shown over here. And this is a picture. It shows that when we're placing the beads directly on the moly target, and you see that's very, it looks all the same. That's a good indication that it's very robust and reproducible. And indeed, if you're looking at the CV, over here is better than 5%, and this is exactly what the clinical chemistry laboratory and the FDA and all the other organizations would like to see. So one result here. These are real samples, and actually like not only from fresh frozen. We have developed this for formalin-fixed, paraffin-embedded tissues, which is not easy to deal with, and I think Tom will talk about this. These are the results that we analyzed from the tissue part. The pathologist said like this is normal and this is cancer. And what we figured out is that the expression level in this patient from AKT1 from normal to cancer goes up, and the phosphorylation as well. This is what we expected. But here in this patient, the expression level goes down, but the phosphorylation level goes up. Now we need to correlate this to the clinical outcome. So this is what we're doing right now, and this is pretty much the end of my talk. The 50 minutes are up. I would like to thank the people at UVic Genomics Proteomics Center, in particular my student Robert. Then the folks from McGill, these are surgeons and clinical scientists. And this person from Germany, he really can make these antibodies what we need to pull out the specific peptides. And our funders, in particular, Genome BC and Genome Canada. And I would like to thank you for your attention.
Thank you very much, Chistoph. So the final talk, which will be given by Igor Ardoris, who is a mental coach who has been coaching some of our top athletes in Sweden and also been involved in coaching patients, I think, in the healthcare. Please.
Thank you. Okay, hi. My name is Igor, and my 15 minutes starts now. So my interest in mental training has always been due to my interest in handling difficult situations. So I have some... do you hear me well, by the way? Okay. I have some... oh, thank you. I have some, I don't know, maybe special experiences due to my upbringing. I had... my father had a lot of problem with alcohol, and then when I was 16, 17 years old, civil war started in ex-Yugoslavia, which made me to change environment totally, and also my expectations and understanding of human nature. So here we speak of one kind of cancer, which is, of course, the physical kind, but many times I've been pondering upon the similarities between how bad people can treat each other and create suffering to each other, and how cancer can do the same thing on the individual way, on the individual level. So my interest has always been in handling difficult situations. Now, one thing we can say about our physical reality is that it always changes, and already for a couple of thousands years ago, people noticed what they call for the truth about suffering, which means that no matter how good life we have, there are some facts that we so far cannot avoid, and one of them is that we grow older, and it's not a problem until 30, but then it's not so fun. And the other problem is, of course, that we get sick, and the third is that we die, and no matter who the person is, this is the similar reality. So the interest for me has always been about, if we look at the human being as a combination of mind and body, of course, and I say mind and body, but actually it's mind-body. So if you extract the mind out of the body, then it's corpse, not the body anymore, not a live human being. And of course, if you extract the body, then I don't know what it is, maybe some kind of ghost or something. So as long as we are alive, we have a combination of these two aspects. And one interesting thing about it is, how do they influence each other? So when we speak of mental training, actually, what I would like you already now to start maybe thinking about is that we are always training. We are always doing something, and those kind of behavior that we repeat, consequently, it becomes habit. And this habit starts, of course, guiding my life. And we have some habit about how we act, but we don't act just acting. That there is first and foremost some kind of mental habit that drives my way of behaving. So when I speak of mental training, we can actually divide it in two parts. We have conscious training, where I choose myself to exhibit some kind of practice, where I consequently strive to learn my mind and my body to think or act in a certain way which is beneficial for me. And then we have everything else, where I get trained by circumstances. I will explain it further in a second. Let's see if this one works. Yes, wonderful. Now, this is one interesting acronym, because it's BICE in English, but not so good word in Swedish. But anyway, I keep it because it's the easiest way to remember. And we have four components of self-hypnosis, or which is the basis of mental training. And these components are belief, imagination, conviction, and expectation. And I underline imagination in red color, because actually it's the main factor of these four components. So let me say that when the things we believe, when we believe something, then we have some kind of internal picture what we are believing. How will the future play out? How will the things develop? And then I have this internal presentation. You can almost call it like kind of reality simulation in my mind. And depending on what kind of picture or internal movie I make, if it's happy movie or bad movie or scary movie, my body reacts to that. Already in 1800, there was a guy called Emil Coué who was kind of apothecary, pharmacist. Thank you, pharmacist, and it was not the same as pharmacist today. More of like combination of doctor and chemical person who works with chemistry. And he said that, well, consciously it's quite difficult to produce saliva, but if we imagine that we eat some good meal, then this happens immediately. So there are what we imagine has a immediate effect on our physiology. Imagination, I already mentioned that conviction and expectations are kind of cousins, but when I'm expecting something, especially if I'm convinced that something will happen, then my body already starts adapting to this expectation. So, for example, if I expect that when I'm done with this presentation, somebody will beat me up, and if I really, really expect that, already now my knees start to shake, my voice also becomes shaky, and I'm having a problem to focus on what is the next thing that I'm going to tell about. Now, somebody could tell me, oh, come on, Igor, I mean, it's in 10 minutes. You have 10 minutes to enjoy your life because before you get beaten up. So why are you worrying about that now? It's not logical. But you know, the problem is because my body is always in here and now, but my mind, as a human being, has ability to imagine stuff. Now, what I'm imagining, my body has no choice, but it has to experience it in the now, because body is always in the now. So even though my fear is about the future, I'm experiencing this fear in the now, which in fact creates all these stress reactions, which are usually not very good for my recovery. Now, if I instead expect that I'm going to have a nice piece of cake afterwards, and let's say that I like cake, already now I'm having saliva. I mean, anybody can say, but come on, it's not before in 10 minutes. Immediately the same thing. So you see, we have, our expectations have a direct effect on our physiology. Let's develop this a little bit more. So, conscious mind, the part of our mind that is analytical, slow to react, one activity at a time, controls less than 5% of our body function. It's motor function, so I can move, I can speak, I can propel myself in the room. And it's a short memory. Well, I have this picture of a bouncer because the analytical, logical, critical part, that's the part which has a critical faculty. So if somebody tells me that afterwards I am going to buy 2 kilos of blue apple, this part will no way, there are no blue apple. Forget about it, because this part is logical and it tells me that's not realistic. But if I'm dreaming, when this conscious mind is deeply relaxed, I can have all different kind of bizarre dreams where blue apple is like nothing compared to what I can dream. And I am not questioning that dream because the part of the mind that is able to question is busy being asleep. So the bouncer is out for a lunch. Okay, if we compare now to the subconscious mind, the part of the mind, we can actually, it's not like the isolated part, it's more the part of the whole, and we can define it like with everything that we are not conscious for the moment, that is the subconscious. And I'm using this picture of the iceberg. It's quite classic, but the thing is, why I'm using it as partially to show how much bigger this part is, and partially also to show that these are not two minds that are together, it's just one mind, just different aspects of one whole. And this part cannot, it lacks discernment, ability to critically look at the reality. This part believes in fantasy. And we have the 6 functions of the subconscious. It's a memory bank or a computer. It controls and regulates involuntary functions, physical involuntary. So circulation, elimination, digestion, breathing. I don't need to consciously attend to these to take care of them. The part of my mind is taking care of this without my conscious involvement. This is the seat of our emotions. And this is very interesting because emotions are this accounts for subconscious mind's domination of the conscious mind, because the emotions, our emotions, govern the strength of our desires. And our desires are what governs our behavior. So one very, very old example is, if you need proof that your conscious mind has quite difficult to override subconscious, I will explain the following experiment. So if you have when you pit all your logic and willpower against your subconscious, in this case, your imagination, which is the next seat of, which is the next part that subconscious is ruling over. So if you lay a 10-foot plank of wood, which is 3 meters, and which is 1 foot wide, 30 centimeters, on the ground, and walk the length of this plank, you will find this quite easy because your subconscious mind is not expecting that this would be a problem. It's not a difficult balancing act, so everybody can do it. And you can do it a couple of times. And now, if you lay the same plank from roof to roof between 2 10-story buildings, and now try the same thing. If you try to walk across from one roof to the other, now subconsciously we are imagining what will happen if I fall. Also, I'm expecting there is a big risk that I will fall. And it doesn't matter how much consciously I'm trying to convince myself, well, you did it so many times before, you know you can do it. When we have a conflict between conscious mind and imagination, expectation, and belief, you can guess who will win. So this is the problem when people try to lose weight and they follow weight loss program, but they are still imagining how good it would be with junk food. Or people are trying to quit smoking, but they are still imagining how good it would be with a cigarette after a meal. So that's why when we use will, conscious will, it requires a lot of energy, but it's only a matter of time when we are low on energy, when the old habits will come back. Now, why am I telling you this? This is quite interesting when it comes to getting well or getting through the process of medication or being treated by doctors. And I have quite a lot of examples which are about this, but I will tell you 2. The first one is about a friend of mine who is more than 75 years old now, but for more than 30 years ago, he got Parkinson's disease. Actually, 40 years ago. And he just noticed that his body is not following the ideas from his mind, or the messages from his mind. And when he got his diagnosis, it was dramatically simple, because he went to doctor and doctor told him to sit down and do like this with his hands. And after a while, one of the hands didn't follow the other. And the doctor told him, well, you have Parkinson. And he said, okay, what does it mean? Well, your body will slowly stop functioning, and within 5 years, you will probably sit in a wheelchair. Remember, this was 40 years ago. I don't think people speak like this now in medical circles, but this was his reality. And well, he went home. It took him 6 months to tell to his wife about this, and a couple of years before he told his daughters. And the interesting thing here, because I have to make the story short, is that he noticed within a couple of weeks that his right arm, that was the biggest problem, that was the first symptom of which made him to look it up. He couldn't move it more. And afterward he said, well, I am an engineer by profession, and as an engineer, I'm more happy to be exactly wrong than to be almost right. So in my mind, I calculated if I'm going to sit in a wheelchair within 5 years, then maybe already now, within a couple of months, this right arm should stop functioning. So when he was, they have a house, and he was having some cakes and coffee on his way up to the upper floor, and he was walking very slowly because he was having difficulty to balancing all this stuff with only one arm. And when he was maybe halfway up, his wife told him, "And come on, take it with both arms. Don't be such a baby or something." And he got really angry. So he turned back to her and he told her, his name is Igor, he lives quite close here, and he told her, "Don't you understand that I can't move it?" And then he did like this when he yelled at her. And she told him, "Well, you're already moving it now." "Yes, I am." And he went down and they hugged, and he told me, not only me, he had a lecture. And he told, well, after this really made him so conscious about how much imagination, expectation has, how big impact it had on his state of being, both mentally and physically. So he started every morning to imagine himself, before he goes up, skiing with his grandchildren. Now, at that time, his girls didn't even have a boyfriends. They were, at least what he knew, they were in their adolescence. So 10 years later, when his oldest told him that she is pregnant, he started to cry. And she got really sad and angry because she thought that he was sad when he heard that she is pregnant. But he said, "No, these are happy tears because I've been skiing with this child for 10 years every morning." And this guy, he still has Parkinson. I mean, this is not a movie, it's for real. And but he's far away from wheelchair. In 2008, we were in China together, and he gave his lecture and I gave mine. And I mean, not only he went there, he needed to go and lay down much more often than the other people, but the rest he took care as everything as everybody else. Now let me give you another example, even now because I have to finish. But I've been waiting for an hour longer, so I will take that liberty. My best friend, he got lymphoma for 10 years ago, and I was with him every day. And they didn't know he also had a pain in his back and he couldn't sleep, so that's why he looked for help. And at the end, when they diagnosed lymphoma, he took it very well. I was there as his closest relative. And afterwards, I went home and called him in the evening to tell him good night. And he sounded like from grave. He sounded really, really in bad shape. And I mean, when you know a person so well, then you can notice in their voice that something is horrible. And I told him, "Well, what's wrong? What happened?" And he said, "Well, I don't know if it's worth fighting for. I mean, what the hell? What happened? I mean, I was there when you got diagnosis a couple of hours ago. Why are you reacting like this?" Well, what happened? We have a mutual friend who is a doctor. At that time, he was a young doctor. This doctor went to visit our friend who got diagnosed by lymphoma. And when he heard about that he's going to go through chemo, so he told him everything he can expect about chemo. And he told him, "You will feel sick, you will be depressed, you will have pain, you won't be able to swallow, you won't be able to graze up," and you can imagine what everything, worst case scenario, side effects of chemo. Well, I can tell you, I was with him every day during, he got his chemo, I believe, every second week because it was very aggressive and it spread to his spine and his lungs. But he was never so bad. He was never in such a bad shape as when he was expecting, hearing it from a friend and hearing from a person who is a doctor, because his analytic guard was down. So he was experiencing already all the worst side effects even before they started to give him chemo. So I asked him, "Okay, what's the point?" Because he said, "I don't know if it's worth fighting for." Why should I torture myself? And I asked him, okay, is there anything worth fighting for? And he told me, well, there are two things. One is you are still young. This was 10 years ago. So hopefully you need me more in your life, he said. And the other thing is, I don't want my mother to experience burying me, putting me in grave. So those are the two reasons I want to fight for. And then we decided, okay, let's focus on that. Now he was lucky and survived. He got new cancer last year. But now he's free from cancer again. And this was the HPV virus, something completely different. So relax.
Now there is a question: if you have a person who is really, really sick and trying to survive and going through the treatment, and how do they think way forward? It's not always that easy when it comes to cancer. So this is a big question. And there are people who've been doing research in this area, and I will just move forward to this guy, who is Herbert Benson, and this is a cardiologist from Harvard University. The guy already in the late 60s wrote an article about teaching monkeys to decrease the level of their blood pressure. So he's the one who coined the expression of relaxation response, which is actually a scientific name for meditation. And according to Dr. Benson, this is the opposite of the stress. We have fight or flight. Walter Bradford Cannon, who coined the expression more than 100 years ago. And the Benson, for 50 years ago, there is like a counter lever, which is called relaxation response. And what it's all about is breaking the train of everyday thinking by repetition and disregarding of the other thoughts when they come to mind. Now, it's difficult for me to imagine that I will be healthy because I don't know. I don't know. So every time when I start to convince myself, well, I will be healthy, there is kind of worm in my mind that tells me maybe you won't. And this is scary. But one way how to handle this is that when I stop the train of everyday thinking, and here is the method, it's a very simple method actually. Pick a focus word, short phrase, or breathing, or whatever, something simple that you return your mind to, where you keep your focus. Sit quiet in a comfortable position with your eyes closed, relax your muscles. There is actually a connection between relaxation and focus. Many people instinctively, when we try to focus, we start tensing ourselves. But there is actually a very big connection between relaxation and concentration, because when I relax, I also start to creating some distance between me and everything around me, but also other thoughts inside of me. And breathe slowly and naturally. As you exhale, repeat your focus word or phrase silently for yourself. It can be peace, one, whatever. If you're religious, doesn't matter what kind of religion, you can use the phrase that you feel is giving you calm and peace. This is the thing. When other thoughts come to mind, simply say to yourself, oh well, and then go back, gently return to the repetition. Continue this 10 to 20 minutes. This kind of practice helps people to experience that they don't need to identify themselves with thoughts or feelings that they have for the moment, but it creates some kind of distance. So the difference becomes, I am the one who is having this thought or this feeling, not the feeling is having me. So when I can feel this, when I can create this distance, and when I can relax, then I can play with what happened if I survive? What would I do? And then you start playing with this idea. But your body is reacting to that. And this is not like a magic formula. You start expecting good things and then you survive. I mean, it would be great if it would be like that. But the thing here is that let's look at panic attacks. People start getting panic attacks when they imagine getting panic attack. So when I work with people who are scared of flying, when they imagine to being locked inside of airplane a couple of 1,000 meters up in the air, and they can't leave until before the plane lands, but they feel like claustrophobic, just imagining this, they start experiencing the effect of having panic attack, because the part that is governing imagination is also governing the physical. So this helps people on individual level to understand, well, I am not my thoughts. Am I reacting now to the reality of the situation, or am I reacting to my thoughts about this situation? And this helps to relax with the reality of the situation. This is the whole thing. And in everyday life we say this is like creating open mind. The mind is open to possibilities, but also able to stay with the situation as it is right now, instead of thoughts about the situation. And of course, we can put this on one more level, and this is metaphysical level. And metaphysical level is all about imagination. I mean, every religion is about belief and imagination, but I mean, it helped a lot of people. It killed also a lot of people, depending on how you apply your imagination and expectations. Do you want to convince others, or do you use it as a tool for your own personal well-being? And on a metaphysical level, at least in the Far East, they have this way of looking at the mind, instead of only looking at it as electrical activity in the brain, which will disappear when we die, so it's nothing more than electrical activity. They look at each mind as a part of one cosmic mind, so as small quanta. So when you come in contact with a part of yourself that is quiet, so there is nothing there that you can put a word on, but there is still something. It's not empty. So it's nothing, no thing, no physical thing that you can describe, but there is some presence of you being conscious. And when you spend time with that part of yourself that you didn't create with your fantasy, you just can notice it by being present in the moment, then you start noticing, well, if my body starts changing, my ideas, my feelings, everything is in flux, but this part, under everything, it seems like it stays. And of course, it's only about, you can choose how you interpret it, but, and what is the true? Is it only electrical activity, or is it the part of the big universal mind that everybody is sharing together? I don't know, but I know what feels best. And this is the part that is interesting from mental training perspective. What is your subjective truth? Because we have people who have problem, how do you say, anorexia, anorectic, or whatever. They experience themselves as too fat, even if they are extremely skinny. But this is their personal subjective truth. And the rest of their life is governed by that. If you have people who have a personal subjective truth that they are unlucky in their relationships, no matter, this is the same as example of balancing on the plank. Even if they try hard to have a good relationship, if they are expecting and imagining and convinced that they are no luck in relationships, then they start behaving like that. So we have science, which is extremely important for curing people for real, and also for synchronizing different parts of society, so we don't get governed by subjective stuff, but by stuff that is really trustworthy. That's why science is extremely important. But at the same time, by respecting and accepting and loving science, we also need to understand that as human beings, we also have some subjective part, and that part, if we experience it with feeling, it really affects us physically, even though it's very subjective. So we need to create balance between that part and the objective part, and make them work together. I prepared much more pictures, but my time is out. Thank you for your interest.
There's been a lot of discussions where patients have presented their own experience, and also where relatives have presented their ways of handling this situation and into the future. There are children involved. So you have all the generations straight through. You have had a lot of scientists giving more or less understandable, according to Gary, ways of providing what we're trying to achieve. So with this, we will close this symposium. We would like to thank, as organizers, all the foreigners, everybody who came, all the participants. It was a really magic day. It was much better than we ever expected. So thanks again. Right now we will have a, we go downstairs 2 levels to D13, it's called, in this building. And then we will have some refreshments and also guided tours. So you will see what all these young kids from all over the world are doing every day, from morning till evening. Okay? Thanks a lot.